Okay. I have already mailed you the notes for today's class and also a small YouTube link and write up from Promega Corp, which is one of the very premier biotechnology company which is involved in making a lot of reagents for molecular biology. Okay, so they have this technical support team. They had given out a handout on how this blue white selection works. So hopefully between the class, that video, short video, and your this thing handout, you will be able to gather a lot of things. You will be able to put together good this thing, information on how exactly cloning works. And by end of the class, we will have a short quiz. I will be sharing the MS Teams form. The link I will share. This is a blind quiz, right? So you don't need to write your name, nothing. So we will get to know overall, how the module has progressed. Okay, so we are moving into a totally into new territory, right? Unlike the previous 4 semesters, things are a little different from this semester. I hope you guys are able to perceive that. Okay, let me start my presentation. We are going to rehearse, or we are going to basically go back to the gene cloning techniques. Okay, we are going to look at few methods today. Typically when we talked about gene cloning, even in my previous class, I said that the most common thing that happens is people start using certain enzymes and certain techniques, but there are a few other neat techniques that have kind of replaced the classical way of cloning. Okay, so we're going to look at a few of those things. So we're going to basically look at methods and the mechanisms. Okay, so is my slide visible?
Yes, sir.
Yes,
All
sir.
right, good, good, good. Okay, so if you look at it, this picture shows you a typical cloning vector. Okay, you have, cloning vectors, as we saw in the previous class. Cloning vectors are meant for cloning your piece of DNA and make enough copies of it so that you can do analysis. You can do give it to other researchers who may be interested in it, or as a backup. There are several reasons why you want to make a clone, and this is a typical cloning vector. And we had seen in the previous class, a cloning vector typically has three very very important components. One is this origin of replication. That is what makes the vector or the gene cloning vector replicate inside the bacterial cell. Okay, so this, as I said, is a prokaryotic cloning vector. So you have, there was a time when people would do cloning in yeast, so they will have a eukaryotic cloning vector, but now things have changed. Now people do what is called as shuttle vectors. Okay, so you have things that can work in both yeast and in E. coli. Okay, so we are not going to worry about that for the time being in today's class. So today's class we are going to look at a prokaryotic, typical prokaryotic cloning vector. This will have three components. One is the origin of replication, so that this plasmid can replicate inside the E. coli. It has a antibiotic resistance selection marker. In this case, this is ampicillin resistance, right? AP, and the R is put as this thing superscript. So that actually means resistance. So when there is no R, that means it is sensitive to it. Okay. So typically, because this carries the beta lactamase gene, which metabolizes the ampicillin and confers resistance to ampicillin. Okay. And then you have a small region which is called as lag-Z alpha. Okay. Today's class we are going to focus on what this lag-Z alpha fragment does. Okay. Within this lag-Z alpha fragment, you have what is called as the multiple cloning site. Okay. Some people call it as polylinkers. Some people call it as multiple cloning site. Okay. Now, what is this multiple cloning site or polylinker site? It is essentially a collection of, okay, series of restriction enzymes. For example, in this case, you can see the multiple cloning site has sites for ECOR1, SAC1, KPN1, SMA1, BAMH1, XBA1, SAL1, PST1, SPH1, Hindi3, right? So this is the thing for PUC19. This is from a company called New England Biolabs. Okay. Now NEB also has its own version of this plasmid, which is little improved, and it carries whole bunch of additional restriction enzymes. Okay, so this is probably little bigger in size. So the size of this plasmid is around 2,686 base pairs, or typically we call it as 2.6 kilo base pairs. Okay, 2.6 kilo base pairs, and it has a lag Z. Okay, and the promoter is somewhere here, and it is driven in this direction. You can actually see each of them have a direction to it, right? The origin of replication, the promoter is in this way, and it drives the gene product towards this. Lag Z is this way. Ampicillin resistance is from this end. It starts, and the direction of transcription is towards this end. Okay, so I hope you understand what the direction of these arrows mean. Does that did I make it clear?
Sir, can you explain just the direction part once more?
Yeah. Somebody had some other question. All right. So the direction essentially means that there is a promoter that is driving the LAG-Z gene from this towards this direction. Okay. Ampicillin resistance. This promoter is somewhere here, and it is driving towards this direction. And origin of replication, the promoter is downstream, and this thing is being driven towards in this direction, basically. Okay, that is what it means. Not a big deal. Not a this thing to worry. The orientation of these can actually change depending on where your promoter is. Suppose the promoter gets here, then the orientation of this will change. But doesn't matter. All it means is, in this case, they are all in the same orientation. They are all in the same orientation. But you can have vectors that can have this orientation little different. Okay, so let's not worry about it. Now, within this multiple cloning site, you see all these different restriction enzymes, and you will be cloning your gene of interest in one of these places, right? And that is what we're going to look at it, okay? And this is a typical prokaryotic expression vector, okay? Do you see a difference between this vector, which is 2.6 kilobase pairs, and this vector, which is 5.4 kilobase pairs? Okay. Now this again has an origin of replication, right? This has an origin of replication. It has a kanamycin resistance marker. So unlike the previous one, which had ampicillin, this vector called PET27B as a kanamycin resistance marker. Okay, it has an F1 origin, okay, which essentially helps in making this a single stranded DNA. It has a lag repressor region, okay, which you don't see this lag I. You don't see this lag I in the previous one, but you will see here. And then you have a short multiple cloning site where you can clone your gene of interest, okay. And this is another vector called PET21A. The only difference between PET27B and PET21A is that here it is kanamycin resistance and here it is ampicillin resistance. Otherwise, both of them look very much the same. The T7 polymerase promoter is present here. We will see this in detail when we talk about the expression of foreign proteins in E. coli. Okay, so we'll not worry too much about at this point in time, but just to tell that between a typical prokaryotic cloning vector like PUC 18 or 19, okay, and an expression vector like PET27B, the first thing that strikes you is the size of the plasmids are little bigger. Expression plasmids per se are little bigger compared to the cloning plasmids, and they have additional modules like LAC-I, which is a repressor, and F1 origin, the origin of replication for making single-stranded DNA fragments. it is necessary for certain reasons, typically for sequencing. Okay, but there are also other reasons why you would like to have a F1 origin of replication. Okay, so is the difference between a cloning vector and an expression vector clear? Can we move to the next slide?
Yes, sir.
Yes, sir.
Okay, so that was the prokaryotic expression vector. This is the eukaryotic expression vector. So this is an yeast expression vector. You can actually now see that the modules are increased. You know, you have additional modules. This is a pgal1 promoter, which is going to drive your gene of interest, and you have a transcriptional terminator from the cyclin genes. Okay, I hope you remember your cyclin genes from your cell division classes, right? And then you have this PUC origin, which is essential for this plasmid to replicate in E. coli. So this is a typical example of a shuttle vector that can grow in E. coli, that can also grow in yeast. Okay, and yeast has its own 2-micron origin of replication. It has a selection marker. See, previously we used ampicillin, kanamycin as selection marker. Here, we're going to use URA3 as a selection marker, which is responsible for the synthesis of uridine. So you will be transformed, your cells will be not be able to synthesize uridine. And the moment this plasmid gets into that cell, they will start synthesizing uridine. Okay, so this is for yeast. And this is for mammalian cells. There are times when we need to express proteins in bacterial cells. Sometimes we need to express in yeast. Sometimes we need to express in mammalian cell. And this is a typical mammalian cell. Again, this is a typical shuttle vector because we cannot be making plasmids from mammalian cells. So we typically do all this. Many of these things are done in E. coli, and then we introduce this plasmid into the mammalian cell. So you can actually see this has a CMV promoter, which is called cytomegalovirus. I'm sure all of you must be aware of it. And then it has a small intervening sequence, which is like, remember all your eukaryotic genes have introns, exons, and all that. So it is somewhat like it kind of mimics that structure. Okay. And then you have what is called as MCSA and MCSB. MCSA and MCSB, if you remember, this is this one, multiple cloning site, right? So this has a multiple cloning site at 2 different regions. Each of them carry a different kind of a restriction enzyme site. And then you have what is called as internal ribosome entry site, which is necessary for optimal translation of the protein. Okay. And then it has an SV40 origin of replication so that this can replicate in mammalian cells. Okay, and this has neomycin resistance, which is used for selection in mammalian cells, and it has ampicillin resistance for selection in E. coli cells. Because this is a shuttle vector, right? It has 2 selection markers. One is ampicillin resistance, one is neomycin resistance. In the case of this, you have this as a selection marker in yeast and this as a selection marker in E. coli. Do you get the 2 selection markers in PS2 and mammalian vectors? Just to show you the structure of these plasmids. Okay. Can we go?
Yes, sir.
All right. So when we talk about cloning, right? Let's say this is your, you have discovered a novel organism, and you want to, isolate, say, for example, some restriction enzyme gene from that. Okay, you could probably be using isolating the genomic DNA from that organism, and you will be subjecting it to restriction enzyme digestion. It could be ECOR1, it could be BAMH, it could be any of the restriction enzyme. Just taking an example of ECOR1 here. Okay, so when you digest your genomic DNA with ECOR1, you're going to generate hundreds and hundreds of fragments, several fragments you're going to get. Some will be short, some will be long. You can see, some are short, some are long, some are medium size. You will have different fragments of different length. And then this is going to be your cloning vector, which you're going to cut with ECOR1, right? And then you're going to add these 2 fragments, right? Your genomic DNA from this and your vector. A circular vector, once you cut with restriction enzyme, it is going to become linear. It's like remember your rubber bands. You cut it in the middle anywhere, you cut it becomes linear, right? A circular thing becomes linear. It's something like that. Okay, now when you add ligase to these 2 mixture, There are typically three reaction products that are possible. One, by intramolecular reaction, these guys can circularize among themselves. This can circularize among themselves, right? Is that possible? Is that right?
Sir, I didn't understand your question.
When you do ligation, right? When you do ligation, the ligase doesn't look at, it only looks at DNA. It doesn't say, oh, this is vector and this is my insert and I have to ligate these two, right? So typically when you add ligase, you have two kinds of reaction. One is called as the intramolecular reaction and one is called as the intermolecular reaction. In an intramolecular reaction, these DNA fragments can ligate back to themselves. Similarly, this plasmid vector can ligate back to itself. Right? And then in your intermolecular reaction, you have one fragment from this and one of the, your genomic DNA and one of your vector DNA get ligated and then you have this new product. Is that clear to you?
Yes, that's clear.
Okay.
Yes, sir.
Now, when you do your transformation, bacterial transformation, cells can take up any one of these products, right? It can take up this circular DNA, it can take up this circular plasmid DNA, or it can take up this recombinant DNA. So this is now a recombinant DNA, right? Okay. Now, in this case, the cells die. Oh, in these two cases, the cells survive. Any idea why cells die in this case? In the case of genomic fragment getting ligated back on itself, the cells do take it up. It's not that the cells don't take up, they take up and they die. Why?
So I'm just guessing here. Is it because it's just a fragment, so it's not able to perform the way it's generally supposed to?
Sort of. You are somewhere in the middle. So typically, if you look at, just compare this versus this. Compare this versus this. Okay. What does this vector have? This will have ampicillin resistance, kanamycin resistance, something will be there, right? And these cells are going to be plated on a plate with ampicillin or kanamycin, right? Do these fragments have the ampicillin resistance or kanamycin resistance? They don't have, right? So they all will die. Okay. This plasmid, when it recircularizes, it will still have the ampicillin resistance gene. So they all will survive. Similarly, this one, which has received a copy of this, foreign DNA, where it got ligated, this also carries a functional ampicillin resistance gene or kanamycin resistance gene. So the cells will survive, right? So did you get why some cells will die, why some will survive?
Yes, sir.
Yes, sir.
Okay, so typically at the molecular level, how does ligation work? Remember I said ligation is nothing but the formation of a phosphodiester bond between the 5' phosphate and the 3' hydroxyl, right? So when your vector DNA and your insert, when they come together by complementary base pairing, these two have a hydroxyl group in the vector and you have a phosphate group in the insert. Similarly, you will have a hydroxyl group in the insert and a phosphate group in the vector. Typically, what does the ligase do? It joins these two together and results in the formation of a phosphodiester bond. Okay, so it becomes one single linear molecule. Okay, we will not worry about this dephosphorylated vector. We'll come back and redo this at a later time point. But essentially, you are going to create a molecule which has phosphodiester bond in one, and then you will have one in which you may not have it, but when it gets into E. coli, the E. coli repairs it. Okay, the E. coli repairs it. So let's not worry about it. But there are reasons why you will use this kind of a thing. As I said, this intermolecular reactions. So there is no way you can prevent this intramolecular reaction from happening. Okay. But there are some neat tricks by which you could prevent this, and you can essentially favor everything to go in this direction. Okay. And how do you do that? Basically, remember, you have a phosphate group and the ligase only looks at a phosphate and the hydroxyl group. So essentially, when you do what is called as a dephosphorylated vector, this phosphate group is removed. Okay, this phosphate group is removed, and by removal of this phosphate group, typically you are able to bring down your this thing intermolecular reaction by more than 99%. So essentially you're forcing, because when you have vector with two hydroxyl groups, the ligase can never work together. So you're essentially forcing the vector and the insert to ligate with each other. Okay. So we will talk about this more when we talk about enzymes in molecular biology. Okay. Now there are other ways by which you could do very easily cloning. Okay. One is to have the use of an enzyme called terminal transferases. Okay. So you basically take a cloning vector, okay, and then use a terminal transferase enzyme and use any of the any one, okay, DGTP, for example. In this case, DGTP is used, and this typically adds at the 3' terminal of the vector a series of G's, because only G is available here. It adds a series of G's. Okay, so that's why it is called as a terminal transferase. It adds only at the 3' end, not at the 5' end. It adds only at the 3' end. On both the 3' end, it adds a few G's. Okay, you need to optimize this. It adds a few G's. Similarly, you can take your insert, okay, and then add DCTP, which will be complementary to this, right? So it adds a few C on the 3' end. Now you can mix and anneal them. You can mix them, and they will automatically, by complementary base pairing, they will get annealed, right? And then you can transform this mixture into E. coli, and within E. coli, so these are nicks. These are not joined together. The E. coli enzyme will join this. Okay, so this is called as homopolymeric tailing. Okay, so this also facilitates cloning. Okay. Ligase by far, ligase is the most commonly used, but in the beginning, people also use this process called homopolymeric tailing. Okay. Then another one we saw briefly last time, that when you do a PCR, when you amplify any gene by PCR, the tag polymerase has this ability to add one A, at the 3' end of every product, you will have a single A, okay, single adenine nucleotide, okay. So typically what happens is, almost all the PCR products will have a single A, especially when you have used tag DNA polymerase. Not all the DNA polymerases, but tag DNA polymerase has this ability to add A independent of the sequence. So it doesn't look at what the sequence is here. There's nothing here, but it adds a single A nucleotide at the 3' end. Okay. Similarly, you can have your cloning vector, right, which will have a T at the terminal. Okay. How do you generate this T? You can simply generate this T by using the previous process of terminal transferase. Instead of DGTP, you will be adding DTTP, right? So you have only T. You have thymidine. Okay, it has a single T. You can have multiple T's. There are ways by which you can generate this very simply by using certain restriction enzymes. You could use terminal transferases, right? You can use certain nucleases, so that you have to optimize it so that it will generate one single T. Okay. Now you can mix these two, and by complementary base pairing, this will get have this thing, and this mixture can now be transformed into E. coli, and E. coli will take over the rest of this thing. Okay, so these are some methods. You saw homopolymeric tailing, you saw the TA cloning. So this is called as TA cloning. Okay, so this is the T and this is the A, so it is called as TA cloning. Another one of the most commonly used methods in recent years is the use of topoisomerases. Typically, in molecular biology, when people thought about cloning, everybody thought only about ligase. But, as usual, there are people who have this ability to think outside the box. And, there was this company called, I think it used to be called Strategene. I'm not sure whether it is existing anymore. But they came up with this neat, not only Strategene, but several other companies came up with this neat trick of using topoisomerases. Okay, when you do the ligase mediated cloning, it generally takes a long time. You need to incubate with ligase at 4 degrees centigrade for almost 24 hours, anywhere between 12 to 24 hours. Okay, but the beauty with topoisomerase is that it takes only 5 minutes. By end of 5 minutes, your ligation is completed. Okay, so basically topoisomerase, when you have a double-stranded DNA, it cuts, it makes a single nick. Okay, and then the second strand is made to pass through this nick. The enzyme is actually holding on to the DNA. Okay, and then it eventually joins these two. It joins these two. Okay, so it is essentially like a intermolecular reaction. It's like two different fragments. Okay, and one of them is getting nicked and then it is getting sealed. So people use this property. So this is fundamental property of topoisomerase. It's not that people didn't know it. They knew this is a fundamental property of DNA topoisomerase. So they used this. Okay. So somebody said, why don't I try doing it with my vector and my gene of interest? Will it work? And it worked. And it worked in a beautiful way. So this now you have these are called as topo cloning vectors. So in this case, I have shown a topo TA cloning vector. You can also have simple topo cloning vectors. Okay, so in this case, it's a PCR product which has a terminal A. Okay, and then you have a topo linked this thing. Vectors are available. You can incubate these two products at the end of your PCR. Okay, this is commercially available. This is commercially available. Okay, with the topoisomerase enzyme linked to the phosphate group. Okay, and you can actually mix these two, and in a matter of 5 minutes, this entire process is completed, and you can transform E. coli the same day. Okay, that is the beauty of having this topoisomerase cloning. Okay, and so coming back to our this thing, when you clone, the vector can ligate back on itself. You can also have recombinant DNA. Those are the cells that take up the this itself. Only your gene of interest, they're going to die. So you will not see them on your petri dish, right? You will see two kinds of bacterial colonies on your petri dish. One will be white in color, the other will be bluish in color. Okay, this essentially tells you that you have your cloning, is it a success or not? The more you see white colonies, that means they are having some kind of a recombinant DNA. It could be, it's probably your gene of interest, or it could be something else. Okay, in all likelihood, a white colony represents a recombinant DNA, something that has a vector and your gene of interest. Okay, and this is the vector that religated back on itself. So it is going to give you a bluish color. So how does this happen? Why is it that these are able to give you blue color and this is going to give you white color? That is what we are going to look at. And this process is called as alpha complementation. If you remember, if you come back and look at this particular vector, which has a lag Z fragment. Okay, so this lag Z gene, which codes for beta-galactosidase in E. coli, is predicted to have about 1023 amino acids. If protein is 1000 amino acids long, how long will the gene be? Any idea? If I say I give you a protein and I say this has 1000 amino acids, how long will the gene be? How big the gene will be? Can we predict? How many codons are needed for coding one amino acid?
So you'll need three.
Three, right?
Yeah.
So one amino acid corresponds to three codons or three nucleotides, right? So that means if it is 1000 amino acid, the gene will be 3 times that size. So it will be 3000 nucleotides long. Okay. Okay. Given that this is about 1023 amino acids, okay, The beta-galactosidase has a molecular weight of 116 kilodalton. Okay, on an average, when somebody says my protein is say 100 amino acids, you can multiply that on an average with 110, and you will get an approximate molecular weight. It's not exact; it's an approximate molecular. After comparing, many different proteins, they have come with the general formula that when it says 50 amino acids, 100 amino acids, 200 amino acids, you multiply that with the value of 110, you will get an approximate molecular weight of a protein. Okay. Anyway, so coming back, this beta-galactosidase has an amino terminal pair, okay, and this is called as the alpha domain, okay, and it is essentially from the first amino acid to the 59th amino acid, okay, and the rest is called as the omega domain, which is the carboxy terminal, okay. So you have N terminal and a C terminal. The first 59 amino acids of the N terminal is called as the alpha domain, and the remaining thing is called as the omega domain. Okay, E. coli strains like DH5 alpha, Xolin blue, so many different strains that are used for cloning. Okay, they have this part of the lag Z deleted in it. Okay, the alpha domain is deleted in them. Okay, so they carry the entire omega domain, but the alpha domain is deleted in them by genetic engineering means. Okay, now when you use these cells, right, and this plasmid, what happens is you have a functional complementation. Okay, the lag Z peptide, the alpha peptide that is produced from here, okay, and the omega peptide that is produced by the host, they interact with each other, okay, and that results in the functional beta galactosidase. Okay, this is called as trans complementation. It is also called as alpha more commonly it is called as alpha complementation, but in a scientific way it is called as trans complementation. Okay, is that clear?
Yes, sir.
Okay.
Yes, sir.
All right. So how does alpha complementation work, and what is the advantage? Okay, so the plasmid encodes the N-terminal part of the beta-galactosidase, which we called as alpha fragment. The host bacterial cell encodes a C-terminal, which is called as the omega fragment. Okay, and the beta-galactosidase can function only one and only when both the N-terminal and C-terminal fragments come together. Okay. Now this beta-galactosidase function can be monitored by the use of a chemical called X-gal, which is nothing but 5-bromo 4-chloro 3-indolyl beta-D galactoside. This is called as X-gal. Okay. Now instead of lactose, see, ideal substrate for beta-galactosidase is what? Lactose, right? This is an artificial substrate, okay, which the enzyme takes it up and it breaks down into two products. One is called as 5-bromo 4-chloroindoxy, and when this gets exposed to air, your petri dishes are going to be this thing, right? We are going to keep it. We are not leaving them in anaerobic chamber. We are only leaving it outside, right? Air is permeating, and that converts into 5 prime dibromo 4 prime dichloro indigo, and this is what gives you the blue color, right? So along with ampicillin in your petri dish, we will also be including a small amount of X-gal. We'll be including a tiny amount of X-gal, and whatever has the plasmid, wherever the plasmid has recombined, right, without any your gene of interest, it will give you blue color. And wherever the plasmid, is this laxy gene got interrupted with your gene of interest, the reading frame gets disrupted, and so that results in the formation of a blue colony. Okay, it is not able to metabolize X-gal. Okay, and so it has a functional omega but doesn't have a functional alpha fragment, so it is not able to metabolize X-gal, and so therefore the colony remains white in color. Okay, did that make sense?
Yes, sir, it did.
Okay, So the plasmid DNA encodes the N-terminus end of beta-galactosidase, right, which is the alpha fragment. When you have your foreign DNA in the alpha fragment, okay, the alpha fragment is you lose the functionality. You're not going to produce a functional alpha fragment. In the absence of alpha fragment, the omega domain by itself is not able to function, and therefore there is no beta-galactosidase breakdown. Okay, and so you don't get a blue colony. Otherwise, it will get a blue colony. So that is why in a petri dish, you will have a mixture of both blue and white colonies. Okay, the more the number of white colonies, it essentially means that your ligation has been successful, right? And you have actually favored the reaction moving forward. Okay, you have favored the reaction. There are a few ways by which you can favor the reaction. One we saw was by dephosphorylating the vector so that the vector cannot ligate back on itself. The other thing is to have excess of your genomic DNA. You take the vector and your cloning vector and your genomic DNA in a certain ratio so that the reaction is more favorable towards your vector and your insert recombination than your vector religating back. Okay, so there are few ways by which we can do that. Okay, so it has been shown in typical reactions. We use a ratio of one vector molecule to three your gene of interest. Okay, so when you do that, 3 to 5 times excess is added when the gene of interest. Okay, when that happens, you will typically create a lot of recombinant DNA molecules only. Okay, so that brings us to the end of our lecture. Let's see, we barely have time. Why don't we do the quiz and then we can look at this, reinforcing this concept with a short video. Let me do one thing. Let me share my this thing. Let me share my link in the this thing. It's taking some time. Let's see. Let me give it a try. I had copied it, so I don't know what to make out of this. Oh God. All right. Copy. All right. Give it a try, and then let's see. I have mailed this in your chat. Okay. Have a this thing. See if it is accessible.
Yes, sir.
Yes, sir.
So I'll wait till 12:10. Okay, so we have about 16. There are about 15 questions. Okay, and we have about 16 minutes to go. So we should be able to do it comfortably. I'm going to be here, so if there are any questions, you can check with me.
Excuse me, sir. It's not letting me submit the form.
Manaswini,
Yes, sir.
uh, so did you see your responses being, does it say it is correct, wrong, or something like that?
No, sir. It's not saying anything. I've just
Okay.
like clicked the options, and then after that, the submit button is not lighting up.
Okay, okay. Let me do one thing. Just give me a minute.
Okay.
Maybe I need to do something. Mm. Hopefully, we'll do it better next time. So I'm trying to see how to share this. Okay, let me do one thing. Let me. Manaspini, I have mailed the this thing link.
Yes, sir, I just received it.
See if you could do that and see does it get submitted.
Okay, 1 second. I'll just finish
Okay,
it.
thank you so much.
Excuse me, sir, it's still not letting me submit.
Okay, but you got the flavor for this thing. Maybe it is my mistake, I guess. So I will have to figure out how to get this done. Okay. But you had a flavor for the kind of questions and this thing, right?
Yes, sir, I did.
We will probably discuss very quickly. So is the class everybody done with the quiz? If everybody is done with the quiz, then we can quickly review these things.
Yes, sir.
Okay, let's
Yes.
quickly do that. Give me a minute. So I think this was a fairly simple. I was just trying it out, so I purposely put it simple. But this can be fairly this thing. little more sophisticated, but it's okay. We will start it out this way. So question number 1 says cloning refers to the process of creating an identical copy of genomic DNA, identical copy of cDNA, creating an identical copy of an organism, and all of the above. So the obvious answer is all of the above. We are not referring to gene cloning. We are referring to cloning as a general statement. So all of the above are correct. Okay. And then the second question is cloning vectors are genetically engineered plasmids, carry antibiotic selection marker, carry an ORI region, which is the origin of replication. And the fourth choice was all of the above. So the answer is all of the above. These are actually genetically engineered plasmids which carry antibiotic selection marker and they carry an origin of replication. And then the question number 3 said expression vectors have the following additional features, right? They have a strong promoter for optimal transcription, carry sequences for optimal translation, they carry an antibiotic selection marker, and all of those. So the answer again is all of the above. Okay, I made a few of them continuously all of the above. Just wanted to make sure that there is no set pattern to it, but it turned out to be that way. Okay. Then which of these enzymes are not essential for gene cloning? Restriction enzyme, DNA ligase, DNA polymerase, ribonuclease. So the answer is ribonuclease, which is not necessary for gene cloning. Okay, other things are all used for gene cloning. Which of these organisms carry a plasmid? Bacteria, yeast, malaria parasite, all of the above. Again, the answer is all of the above because in the last class, I said that even malaria plasmids are supposed to carry a plasmid which resembles chloroplast, right? So these are called as plastid. These are called as plastids, okay? So if you had remembered, then you would have known, okay? The DNA ligases are essential during the process of DNA replication, DNA repair, DNA recombination, all of the above process. The answer is all of the above process. It happens during replication, it happens during repair, and even during recombination when the two different fragments recombine, okay, they are joined together by DNA ligase. So the correct answer for question number 6 is again all of the above. Question number 7, 2 micron plasmids are found in bacteria, blue-green algae, yeast, all of the above. The answer is yeast. The first organism to be cloned was a cow, dog, sheep, none of the above. The answer is Dolly the sheep. Then 9, the most commonly used microorganism in the laboratory is E. coli, Saccharomyces cerevisiae, Thermus aquaticus, T. aquaticus, which is where you get your Taq polymerase from. And both 1 and 2. The actual answer is both 1 and 2. But if somebody says somebody has ticked E. coli, I would still be okay, with that. Okay. Then the question number 10, DNA ligases join 5' phosphate with a 3' hydroxyl group. Okay, that is the actual answer. Okay, there is no other answer to it. The other choices are 5' hydroxyl and 3' phosphate. No, that is wrong. Any two hydroxyl group, that is again wrong. None of the above is not the right choice. Okay. Gene cloning can be done using plasmids, cosmids, artificial chromosomes, all of the above. The actual, this thing is all of the above. Okay. Homopolymeric tailing facilitates cloning by complementary base pairing. We saw in today's class that, you have a series of Gs, you have a series of Cs, they all base pair together, and it is because of complementary base pairing, and it is not because of the ligase activity of the terminal transferase. PCR products have a 3' A overhang, 3' T overhang, 5' A overhang, and 5' T overhang. Always remember a piece of DNA with the 5' and 3'. In today's class, we saw that it is the 3' A, not 3' any of the other things, right? It doesn't have anything at the 5'. It's only at the 3'. So it's a 3' A overhang, okay? Ligation with topoisomerase takes 5 minutes, not 24 hours, not overnight. Okay, it takes only about 5 minutes. Okay, and then blue-white selection is because of the activity of which enzyme? It is not because of catalase. It is not because of aconitase. It's because of beta-galactosidase, which we had discussed in detail today. Okay, so these are the responses. Okay, any questions? Anything that you would like to clarify? That's okay if you had, done some mistakes. That's okay. I think we are going to get better on this. Okay. So we will see. Okay. Any questions?
No, sir, but I think nobody in the class can submit it.
Yeah, yeah.
Submit
No,
it.
I
Yes,
I
sir. I
think
was
it is.
also not able
I
to
think it
submit
is
it.
probably a mistake on my part. As I said, I am a tech novice. I don't know. I'm not as tech savvy as you guys are. So I guess if you people were here, you would have helped me. And because you are not here, I will take the help of somebody else and get this done. Okay, but we will do this nevertheless. From now onwards, we'll have a short routine quiz. This basically, helps you to reinforce the concept, right?
Yes, sir. Actually, Google Forms has much less issues than Microsoft Forms.
Acha, I did see. I actually started with Google Forms, okay, and then somebody said, "No, Outlook has its own form. Why didn't you try it?" So I said, "Okay, let me do that." So I did that. Okay, so maybe I
Yes,
need
so
to.
Microsoft has integrated all its function. Like we can even get a scanner inside in Teams, but
Okay,
sometimes Microsoft Forms has this kind of issues. So
अच्छा ठीक है okay, thank you so much, thank you so much, right? So as I said, you guys are this tech generation. I still belong to that old generation. All right. All right, so take care, have a happy weekend, take care, God bless you all.
thank you, sir.
Yeah.
Thank you, sir.
Thank
Okay.
you, sir.
Sure.
Thank you, sir.
Thank you, sir.
Okay, bye.
Thank you, sir.